start the webinar right now. So good morning, everyone. Thank you for taking your time to join this webinar. I'm Cassie, Marketing Specialist from GenScript, and we are the world's leading biotech company with gene synthesis, peptide, protein, antibody, and preclinical drug development service capabilities. So today's topic for the webinar is Evaluate Neutralization Activities for COVID-19 Vaccine and Drug Development. I'm very excited to introduce you to the presenter today, Dr. Yanfeng Li. Dr. Li is the field application scientist in GenScript. She is well trained in cellular biology and immunology with extensive research experience in signaling pathways determining B-cell biology and T-cell functionality. So this webinar will take around 40 minutes. If you have any questions, please feel free to type in the question box that you can see on your screen, and we'll be answering them after the presentation. If we didn't get to answer your questions during the Q&A session, we'll be sure to take down and I will email back to you. This webinar will be recorded and it will be available on our website after the session today. So without further ado, let's welcome Dr. Li to start her presentation. Dr. Li, over to you right now.
Many thanks, Cassie, for your kind introduction. Welcome, everybody. In this webinar, we're going to introduce a new capability from GenScript that we're able to evaluate neutralization activities for COVID-19 vaccine and drug development. Before we deep dive on neutralizing activities for COVID-19 vaccine and drug development conversation, allow me to briefly introduce our company. GenScript has come a long way since we first launched in North America, New Jersey, 2002. We have since also expanded our presence in Asia Pacific, Europe, and America. Not only in physically we expanding our services, we have also developed full-fledged services portfolio from gene, oligo, peptide, protein, antibody, NGS, biologics, catalog products, and also, as we are talking today, in vitro diagnostics. Since we launched Legend Biotech, the cell therapy segment in GenScript, we have seen exponential growth with GenScript going public in 2015 and Legend Biotech IPO recently this year. With a large R&D center located in Nanjing, China, GenScript is well positioned to quickly adapt and develop various of solutions for COVID-19. We are fighting COVID-19 too, SARS-CoV-2 with the researchers globally on the front line, offering an array of solutions for virus detection, vaccine raw material supply, assay development, and QC methods. Among this capability, we are most excited and proud to introduce to you our new capability of neutralization antibody detection method. Our cPass neutralization antibody detection kit was co-developed with Prof. Wang Minfa from Duke NUS Singapore, and recently we also launched our SARS-CoV-2 pseudovirus neutralization assay kit, which is a cell-based neutralization antibody evaluation tool. So these most recent add-on capabilities on neutralization detection will be my focus of presentation today. Unfortunately, until today, as we are speaking now, COVID-19 spread is still rising rapidly around the globe. So this slide summarizes the different test methodologies available. During the acute infection stage, qRT-PCR is still the gold standard in determining if someone is infected with SARS-CoV-2. Serology testing, on the other hand, can reflect recent infection and also long-term infection history. So IgG tests can be the first line of immune response and can offer information on recent acute infection. IgG detection is oftentimes done together with IgN, can supplement information of infection history for longer period of time. So the virus neutralization test can be used last to evaluate herd immunity and detect the functional neutralization antibodies that can block virus to infection the cell. So depending on different purposes and accessibilities, these three tests can be run in parallel to draw the full picture of one's infection status and allow the clinician to formulate the best care protocol for each of the individuals. How does the virus infect the cells? So the mainstream scientists from the globe reconciled that SARS-CoV-2 rely on the RBD domain, the receptor binding domain of the spike protein, to attach to the ACE2 receptor for the virus to infect the human cells. If one's immune system is robust and activated by the viral particles, neutralizing antibodies can be raised to fight off the interaction between the RBD and ACE2, and then the virus cannot infect the cell. So objective of a successful vaccine is oftentimes to elicit strong immune response and potentially induce neutralizing antibodies that can fight off the virus. At GenScript, we are proud to be the hidden hero in contributing to many successful vaccine development worldwide. We were featured in Nature article for being a surprising player in the race for a SARS-CoV-2 vaccine and supporting Novavax in early days of the vaccine design. We are happy to find out that Novavax, as you can see from the bottom left of the figure, is currently under phase 3 evaluation on their protein subunit vaccine candidate. Our last update from WHO on 12th of November 2020 on COVID-19 vaccine landscape, a total of 164 candidate vaccines are in the preclinical evaluation using various of vaccine technologies from protein subunits, non-replicating vector, RNA, inactivated replicating viral vector, DNA, VLP, and so on. So among these, a total of 48 vaccine candidates is now in phase 1 or 2 clinical trials. And most importantly, 11 of such vaccine candidates are already in phase 3 clinical trials with the non-replicating viral vector, inactivated RNA and protein subunit platforms leading this global effect. So I think from time to time, oftentimes it will be advisable. So it's like a stock market. One can always go back to the WHO website, and they do have really timely updates every 2 weeks or so on. They will actually update all the progress of the recent development work. And as you see, in the last 2 weeks' time, we already observed a total of more than 10 candidate vaccines for the preclinical evaluation, and 3 more vaccines in the phase 1 or 2 clinical trials, and 1 more vaccines in phase 3 clinical trials. So this is really like a global race, while companies around the globe are really trying to get their hands into the vaccine, into the market. So how do the FDA guide the vaccine evaluation? As you can see from the FDA development and licensure of vaccines to prevent COVID-19, there are 3 elements of importance to evaluate. Number 1 is the cellular response. Number 2 is the humoral response. And last is the functional immune response. For cellular response, one will be examining the CD8 or CD4 T cell activity upon the vaccine injection. And for the humoral immune response, the aim is to characterize the antigen-specific antibody generation in the vaccine stages. So last but not least, the most critical element of vaccine development is to induce a strong functional immune response and elicit neutralizing antibody production. So wild-type virus or pseudovirus can be the traditional neutralization assay to evaluate the functional immune response on the neutralization antibody titer. In the meanwhile, GenScript is proud to introduce our new methodology, cPass Surrogate Virus Neutralization Test, and hopefully you'll find this add-on capability of help to your vaccine development. So this is a comparison of the various of neutralizing antibody functional assays. On the far left, you can see the conventional VNT assay rely on the live virus and the cell culture to evaluate the virus neutralization activity. So this assay is actually requiring a strict biosafety condition control, biosafety level 3 to perform the experiments due to the presence of live virus. And it takes multiple days to perform the study, requires a high level expertise to read the results. For example, in the context of the plaque reduction neutralization test, one has to manually count the plaque numbers during the different dilution conditions. So because of all these limitations, the live virus VNT assay is actually expensive and not scalable. On the other hand, the pseudovirus neutralization test has been a huge development and is for many of the vaccine companies, because we are relying on pseudovirus to replace the live virus. This assay can be used in BSL-2 level, and it can be ready to use kit to allow the high throughput. So the readout is also depending on a pseudovirus-induced luciferase signaling, so that is much more high throughput compared to manual plaque counting, right? So this pseudovirus is a cell-based functional assay. So these two, live virus and pseudovirus, are both based on cell-based functional assay. What we are going to introduce on the right is the Surrogate Virus Neutralization Test, where we are actually deploying recombinant proteins. So instead of using the whole virus and cell, we actually extracted the RBD domain of the protein and also the ACE2 receptor. So by simplifying this assay, it can be used again in BSL-2, and it's really fast, can be done within 1 hour. And later on, we can also share with you how cPass sVNT test can be automatable and can be done at a super high throughput. And the results from the cPass sVNT test can be much more consistent because we are no longer comparing batch to batch preparation of live virus or cell cultures. Instead, we are only using the biologics production of the RBD protein and ACE2 proteins. So this is a graph to really compare the workflow from the traditional live virus assay to pseudovirus and to surrogate VNT assay. So as you can see, the traditional PRNT, plaque reduction neutralization test, is actually a super intensive workflow, and we wanted to reiterate that the final readout is really based on the manual counting of the number of plaques, so the sensitivity is hugely dependent on the expertise level of the technician. On the other hand, pseudovirus capability is a simplified workflow, can be ready to use. So basically, for the pseudovirus, it can allow the bioluminescence plate reading. So 96 or format of the sample can be read at one time, is offering much higher throughput. So on the other hand, our cPass sVNT assay is automatable, and the assay can be done within an hour. It's a super fast assay to replace the traditional pseudovirus and live virus, or to complement the results, right? So in the way that the cPass sVNT assays rely on the calorimetry plate reader, so this plate reader is actually readily available in majority of the laboratory settings, so making cPass sVNT assay super accessible. This is to share with you in the vaccine trial, how do the pharma industries to evaluate the neutralization activities. So this is a published data. As you can see, Moderna scientists actually used the pseudovirus VNT assay to evaluate a larger cohort sample for their neutralization activities. Only a small subset of the sample was subject to the plaque reduction neutralization test, most likely due to the limitation and accessibility. And oftentimes, I think the pharma industry have to send the serum samples to third laboratory testing facilities to allow running of this experiment for the live virus assay. So as you can see from these two experiments, comparing results were actually achieved in the vaccinated serum, right? And I think with this data, the pseudo-VNT assay can be shown as a reliable alternative to the gold standard plaque reduction neutralization test. imagine these are phase 1 and phase 2 trials. So in the phase 3 trials, right, even for the pseudovirus assay, which takes about 2 days to run, is not going to afford that accessibility to all the clinical samples, while thousands and tens of thousands of vaccinees and the samples will have to be processed. And oftentimes, I believe to analyze all the time points and all the individuals do open new information for the clinical trials, right? Because with a subset of information, one will have to be answering questions. How do I select the samples for pseudovirus or live virus testing? Will that be a bias? Right, so I think the key information here we wanted to show is that if, let's say, for undergoing phase 3 clinical trials with a larger sample set that is beyond the pseudovirus and live virus testing capabilities, our surrogate VNT assay can really offer that fast, automatable solution to analyze the functional aspect of neutralization antibody induction to supplement the assay that we run from the pseudovirus and the live virus plaque reduction neutralization test. So this is to introduce you our data generated to compare the pseudovirus neutralization test against the plaque reduction neutralization test. As you can see, our lead candidate were actually subjected to both the pseudovirus assay and live virus assay on the left. And as you can see, the comparable results were actually obtained between these two assay protocols. On the right, as you can see, right, we performed a study to evaluate the consistency of performance between operator and also inter-assay. Right, oftentimes because of the longer time required, one may have to get multiple operators to run the study. And since the throughput, you have to allow serial dilution of your sample, a single plate will have limited number of samples that you can handle. So looking at inter-operator and inter-assay, precision is one important element for our vaccine companies to validate the assay. So this data suggests that the pseudovirus neutralization kit produced by GenScript was able to deliver consistent performance inter-operator and also inter-assay. So from this slide onwards, I would like to focus my data and clinical sample testing on what we have generated for cPass sVNT kit. So to further reiterate, the sVNT kit can be really that fast, automatable replacement and complement to existing functional assay of plaque reduction neutralization test and pseudovirus neutralization test. So as you can see from this study, actually a total of 66 samples were subject to cPass analysis and also PRNT 90 titer evaluation. So among the 66 samples, 26 of them are confirmed positive by the PRNT 90 titer using a cutoff at 20. And 40 samples were actually negative as evaluated by the live virus PRNT assay. As you can see from the graph on the left, y-axis is actually to indicate the percent of inhibition derived by cPass assay. X-axis is the PRNT IC90 titer that can produce an effective 90% of inhibition. So one can imagine the correlation we have obtained from these two assay protocol comparison is R-squared at 0.9. So a very strong correlation was obtained between the cPass assay and plaque reduction neutralization titer and live virus assay. So I think oftentimes one might imagine, how about the sensitivity and specificity information? So when we are using the PRNT 90 titer cutoff at 20, cPass percent of inhibition cutoff at 20%, you can see that the sensitivity we have achieved was 100%. Specificity which achieved was 95%. So from this experiment, we can confidently derive the information that cPass data actually can predict neutralizing antibody titer with a high correlation to plaque reduction neutralization test. So not only GenScript was able to produce a good validation report for cPass, right? We also have invited researchers around the globe to test the protocol and see how is the performance of the kit in their own laboratory setting. As you can see on the left, we have obtained a result from a total of 154 samples, mixing of human, dogs, cat, and hamster serums, and 47 of them are actually positive and 107 negative. So as you can see, the agreement between the percent inhibition derived from the cPass sVNT assay versus the PRNT 90 titer is highly agreeable, right? And as you can see, negative samples were tested negative for both cPass and PRNT assay, and positive samples were actually positive in cPass as well. So using the PRNT 90 titer cutoff at 10, cPass inhibition cutoff at 20%, we have achieved a very good sensitivity specificity, both at 100%. So this laboratory, this work has been already published. You may refer to more details in this link. So this work is actually produced by a laboratory from Hong Kong. And in addition, our customer from Germany also reported a very good promising data set. So again, the same experiment was actually performed, right? Basically, a number of positive samples and negative samples were subject to both cPass inhibition calculation and also PRNT 50 titer evaluation. Using the cPass inhibition cutoff at 20% and PRNT 50 titer cutoff at 20, as you can see, we again achieved a statistic performance of sensitivity and specificity of both at 100%. So I think both researchers from Hong Kong and Germany was able to deliver a very excellent agreement between the cPass assay and the live virus PRNT assay. And many other works are still ongoing, and we do have a lot more to share if the time allows. So if you're working on the vaccine development, right, you will always wonder, how can we use the sVNT assay to measure the IC50 so that, right, we can produce the same type of results for the for IC50 or IC90 evaluation. So from this experiment, you can see a total of 60 convalescent serum samples were evaluated in both cPass sVNT, live virus assay, and also pseudovirus assay. So as you can see, right, the y-axis and x-axis was actually the log transformation of the neutralization titer for cPass IC50, and also for the x-axis here is to evaluate the neutralization titer for the conventional live virus VNT. So a very good correlation again was achieved when you're looking for the IC50 titer from cPass and the live virus assay. The R-square was achieved at 0.86. Again, when you were trying to compare between the sVNT assay versus the pseudovirus VNT assay, a very good correlation again achieved. R-square was at 0.84. So lastly, the two conventional pseudovirus and live virus assay correlation was found actually slightly lower compared to the level of correlation we have between cPass versus the two traditional assay systems. Correlation was found at R-square of 0.77. Right, so I think with this data, which is published by Prof. 1 in the Nature Biotech paper, one can easily conclude that the sVNT assay can be used to detect the IC50 titer, and the result of which actually correlates very well to the live virus VNT assay and pseudovirus assay. So another question that one might imagine, right, how much time month immune response can sustain for either the post-infection patients or the vaccine samples, right? So basically, these two experiments were performed by investigators from China in this Nature Medicine paper. As you can see, when you look at the binding antibodies, IgG antibody was found to have a marked reduction of 70% during the 2 months of following time. And on the other side, the neutralizing antibody was found to be much more stable, and there was only a drop of 10% of neutralizing antibody reduction. The work produced by Netherlands investigators. So in this study on the right, many of the subjects were actually follow up on this longitudinal study. As you can see, the y-axis is the difference of the antibodies for IgG and also for neutralizing antibodies. And the x-axis is the time differences between the two sampling time points. Basically, the same information was achieved. So you can see that the IgG antibodies, again, dropped significantly with the duration of the time difference between the sampling time points. So it's correlated at 0.46, and you can see the p-value is significantly lower than 0.05. However, on the end, when you look at the neutralization antibody determined by the sVNT cPass kit, right, you can easily see that there is no correlation available, and the p-value was way above 0.5, right? So this paper on the right also indicates that IgG may drop fast, but the neutralizing antibody may be lasting much longer. Taken together, these two pieces of research, right? I think, of course, there are much more papers that are coming up, and we probably are confident to believe that the binding antibodies drops much faster. However, the neutralizing antibodies may sustain longer for the protective immunity. So this is a very interesting article if you haven't seen it before. So basically, in this cruise ship, right, many of the crew members were actually tested for presence of binding antibody, and luckily cPass was used in this study to evaluate the presence of neutralizing antibodies. So 3 of the crew members were found positive for neutralizing antibody presence. And unfortunately, during the trip, there was a viral outbreak, and these 3 crew members who already have the neutralizing antibody titers were shown to be free from viral infection and free from any other symptoms. So I think this is too early to confirm the finding. However, I think the paper do indicate that neutralizing antibodies may be the antibodies that can confer immunity. Many more research are still being conducted until we are clear like how much of neutralizing antibodies may bring the protective immunity. So in addition to use cPass in the vaccine studies, government agencies and hospitals are actually actively using cPass for their serum survey purposes. So in this study, we have evaluate a total of 47 positive samples and a total of 931 negative samples, which results were actually confirmed by RT-PCR. All the 47 samples were collected from 14 days post-infection. So as you can see, right, our agreement from the cPass neutralization antibody presence versus RT-PCR confirmed positive and negative cases are very high. So if you were to calculate the sensitivity, the positive percentage of agreement is 100%. So all 47 RT-PCR confirmed cases have the presence of neutralizing antibody. And the negative percent of agreement is also very strong at 99%. We did see a false positive signal for 8 of the 931 negative samples. However, I think this 100% sensitivity and 99% of specificity is quite strong testimony for government officers and hospitals to start to use cPass for their serum survey studies. And cPass can really help them to deliver in the population who has been previously infected. So this sensitivity and specificity was robust enough to make a lot of policies. And for example, right, in Singapore, cPass was actually part of the serum survey tour for hospitals and MOH to actually identify individuals who may have prior infection history, whether the onset symptom days will be 7 or less than 7 days. So different procedure will be followed by the medical professionals to really allow these individuals who were found positive for cPass reading to stay at home, to go through quarantine, so that I think part of the role cPass is also playing in Singapore in our very effective control of COVID-19. So not only Singapore is adopting cPass for the serial survey purposes, we can see on the left is paper published from MOH Malaysia. So cPass was used as the serology surveillance for neutralizing antibody among asymptom healthcare workers in the healthcare. So in this literature, right, the agreement of the cPass sVNT results were found to be 100% agreeable with samples from the convalescent COVID-19 cases and also negative pre-pandemic samples. So very proud to share with you that within the MOH validation from Malaysia, cPass was actually able to deliver 100% sensitivity and 100% of specificity. So in addition to government laboratories, cPass was also used by private laboratories to adopt cPass as their own testing offers to the public. So Ethos Laboratory in US was also evaluating cPass at the very beginning before they launched their true immune test to the public. As you can see in the test, they have conducted a total of 32 positive RT-PCR samples and 77 negative samples. And sensitivity and specificity was found both at 100% high. So once again, if you are in professional medical service, please take note that the immune response does take time, and we do recommend to take the serum samples at least 8 days beyond the symptom onset to allow maximal sensitivity and specificity in your assay. So one may wonder, the binding antibodies may be an easy alternative to evaluate compared to the neutralization functional assays. So this paper on the left actually indicates the association between the neutralizing antibody titers versus commercial serology binding antibody assays. So 3 of the brands were used in the paper: Roche, Abbott, and Euroimmune, right? And then were found to have a correlation of 0.29, 0.47, and 0.46. In GenScript, we were also able to start a study with a large sample cohort of at least 500 samples for both total S1 antibody and cPass inhibition assay. And as you can see, the correlation was at 0.54, and this correlation between the total S1 versus cPass neutralization antibody presence is much lower than our correlation between cPass versus the live virus PRNT. So again, right, with the sensitivity to compare against RT-PCR confirmed results, the total S1 antibody was found to deliver a 96% of sensitivity. On the other hand, cPass was able to deliver 98% of agreement to the RT-PCR results. So again, when you look at the people who may have high total S1 antibody presence, some of them may not have much of the neutralization function, right? So overall, we wanted to indicate that there may be a poorer correlation between the binding antibody assay versus the neutralization function. And if, let's say, we wanted to really evaluate the neutralization activity of one's immune system, we do recommend to directly work on the neutralization assay protocol. So the sVNT assay can be also used for COVID-19 drug evaluation. In the paper published in Science, investigators was able to identify potential drug targets for screening. In this case, the linoleic acid, which is actually part of our serum components, can inhibit RBD and SCE2 interaction at a level concentration of, I think, 128 nanomolars onwards. So not only sVNT can be used for the small molecule drug screening, I think the biggest application for sVNT was to serve as a good screening tool for the functional therapeutic antibody development and to see which antibody might have higher potency in inhibiting the virus and SCE2 interaction. Many of the discussions are already ongoing, so we do see quite a bit of adopting of sVNT assay for many of our pharmaceutical applications for generating therapeutic antibodies. So this data actually shares with you that cPass, in addition to evaluating the presence of neutralizing antibody and compared to the binding antibodies, right, which actually oftentimes will have to rely on a secondary antibody and is isotype or species dependent. cPass, on the other hand, is actually the neutralizing antibody test that can be free from limitation of different animal models to human trials. So in the graph shown on the left, right, cPass sVNT kit was used to identify neutralizing antibodies present in the immunized rabbit, immunized mouse, and also in the COVID-19 convalescent serum. So sVNT can be used across many of the animal species all the way to human clinical studies. This asset of property really has been very much valued by many of the vaccine companies because you, instead of generating multiple assay validations, you will be free to adopt this single assay, and it can be used across your preclinical evaluation all the way to phase 1, phase 2, and phase 3 clinical trials. On the right, we're showing data that we have generated while we obtained a very little cross-reactivity with the cPass sVNT kit with other viruses like human coronavirus OC43-229E, dengue virus, Zika virus, and MERS, and etc. So as I talked many times in the beginning of our conversation, cPass can be really add-on service that can supplement our existing capabilities using pseudovirus and live virus assay because it can be a fully automatable process compared to a laborious manual process for the live virus and pseudovirus assays. So in this graph, we actually derived a minimal validation report for cPass sVNT kit to perform on a full walkaway automation system. So we have evaluated many of the commercial ELISA automation systems. And in this context, a full plate execution was demonstrated. And as you can see, the sensitivity and specificity for the samples that we have tested in this full 96 plate format is at 96% of sensitivity, 100% on specificity. So as you can see, we purposely chose multiple negative samples, and one spike with neutralizing antibodies, which give a borderline percent of inhibition at just about 20% cut-off line to evaluate like the power to differentiate the positive and negative results. And the third sample was adopted here, which produced about 50% of inhibition. As you can see that negative samples were found to be always negative in this experiment. And the borderline neutralized antibodies spiked sample was found to have a percent of CV at 11%. So cPass results for high-end percentage of inhibition were found to be very reliable. And in this case, we were able to show that the percent of CV for the last sample, which is about 50% of inhibition, is at 5.8%. So in a way, right, I think even compared to the manual protocol, automation systems will offer much more reliable, traceable information. And many of the laboratories really wanted to validate the assay on an automation system. And cPass can comply with major ELISA automation systems to deliver good sensitivity and specificity for your assay. So if there is any other information that you wanted to have one-on-one interaction with us to see whether we have tested your automation system, feel free to contact me. I will have my numbers and email information at the end of this presentation. Again, very proud to present to you that cPass sVNT kit cannot be a possible development technology without our three-party collaboration between Duke and US, GenScript, and A*STAR. So this first-in-the-world SARS-CoV-2 serology test can detect neutralizing antibodies without containment of facility or specimen, can be used for multiple applications, for example, contact tracing, seroprevalence, all the way to longevity of protective immunity, efficacy of different vaccine candidates, etc. So we hope that with all the information we shared above, one can really agree with me that cPass sVNT kit can be used to address unmet needs against COVID-19. Thanks to the high correlation between cPass to plate reduction neutralization test, cPass can be used to study vaccine efficacy of neutralization antibody induction. It can also use to monitor the longevity of the neutralizing antibodies at different time points post-vaccine. On the other side, the cPass can be also adding value to the convalescent plasma therapy to screen for donors and also to look at the recipient neutralization antibody information. So before the transfuse of the serum and plasma, actually is really critical to understand whether the donor has potent neutralizing antibodies and the recipient is probably less presence of neutralizing antibodies, so that it makes sense for plasma therapy to be much more successful. Thanks for the good agreement between RT-PCR results versus the cPass sVNT kit. So the cPass has been used widely for serum surveillance, for government agencies, hospitals, MOH around the globe to identify the infection history of a population and to identify within a population who has been previously infected. When the societies and economy reopens, one will really wonder what is the percentage of herd immunity, and also how many of the individuals who has been undergoing vaccine can be protected from SARS-CoV-2. So with the high correlation to PRNT, cPass sVNT assay can be used to screen community for neutralizing antibodies and determine who has the neutralizing antibodies and save open of the society and economy. Last but not least, cPass can be really used to identify drug candidates to look for the therapeutic neutralizing antibodies and also other small molecules which might have potency in blocking the RBD and SE2 interaction. So this is the graph to show you the product that we have for the cPass sVNT kit and also the PVNT kit. As you can see, cPass sVNT kit adopts a blocking ELISA format, can be done within 1 hour. On PVNT, the pseudovirus assay is a cell-based assay, and both assay can be providing a high sensitivity of the neutralizing antibody detection, and we really recommend you to use the cPass sVNT or PVNT for the vaccine evaluation and drug development and many other purposes like what I have highlighted previously for serum survey and also for the herd immunity. So this is the holistic view for GenScript solutions to SARS-CoV-2. We offer nucleic acid assays, qRT-PCR kit to detect the presence of SARS-CoV-2 particles, serological assays, including the neutralization antibody detection kit we just mentioned, and also many of the RBD spike proteins and IgG or IgM antibodies, depending on functionality and also different level of affinity. And many of these antibodies are really highly looked after for people who are generating new IVD kits and detection methods. In parallel, since China opens the borders and then they actually require the IgM testing to allow individuals to be eligible to go back, right? And we also offer S1 RBD, IgG, and IgM ELISA detection to allow evaluation of recent or prior infection. Last but not least, there is the nuclear peptide, nuclear protein, and also the RBD total antibody ELISA detection kit, also available for your evaluation. For the neutralizing antibodies, we do have the pseudovirus neutralization assays. We have SE2 assay cell lines. We can offer you customized antibody development for your own evaluation, different vaccines, and all the different antibody generation requirements. So we also have in-house produced many reagent antibodies that are derived either by a single cell technology or the hybridomer technologies, many of which are of very high affinity, and we do have presence of multiple antibody pairs that can be really used to leverage for your IVD development needs. So for vaccine development, we have a large peptide libraries, research grade to GMP grade plasmid, and of course our recombinant protein and subunits, recombinant protein capabilities have been really helped the vaccine companies to expedite their internal process to develop their vaccine candidates. Coming to the last slide on the summary, I hope that with the data we have presented here, for scientists who are in the audience and working on vaccine, drug development, or convalescent plasma treatment, you can agree with us that the sVNT and pseudo-VNT can be the kit of choice for functional neutralizing antibody detection because we are able to offer excellent correlation with the live virus VNT, much more simplified protocol, and much more scalable and less expensive to VNT. highly accessible. We don't have to rely on BSL-3 laboratory, and also compatibility with a variety of animal models to humans. So for laboratories who are actually performing serology testing and epidemiological studies, cPass can be the kit of their choice also, because within one kit and one test, we are able to deliver two information. Number one, who has been previously infected. Number two, who has actually a detectable neutralizing antibody presence in the blood. So with that, I would like to thank you for your attention, and I'll be happy to take on questions.
Yeah, thank you, Dr. Lee, for the nice presentation. We'll answer some questions from the floor right now. So the first question is, was the SARS-2 purified? What's the difference between wild type and pseudovirus?
So as you may see, right, the live virus is really the prevalent virus present, and it has high infectability, right? People who are working on the live virus can be very much undergoing a high risk. On the other side, the pseudovirus candidates, I think there are many of those, like VSV, lentivirus vector-based, is able to mimic the core structure of the live virus assay, especially adopting the interaction elements between the virus and the ACE2. Many of the lentivirus vectors may adopt expression of spike protein, especially the RBD domain of the spike protein, which is responsible for viral entry. So I think we may not be able to generalize what are the components for many of the pseudovirus assay that is available in-house or commercial, but basically the pseudovirus will try to mimic what key elements that the live virus may have, and try to come up with this assay to really derive the kinds of similarity during the validation of the pseudovirus versus the live virus. So I hope I answered your question. Casey, maybe we can go to the next.
Okay, sure. So the next question, there's a positive control in the SARS-CoV-2 sVNT kit, which is our kit. The kit does not say what is the positive control, but I think it should be the neutralizing antibodies against RBD. So the question is, what or how strong the anti-RBD neutralizing antibodies titer is in this positive control?
This is a great question. I'm sure that the audience probably has already started to use our cPass sVNT kit. So you're right, indeed, in the cPass protocol, we do have inserted a neutralizing antibody as a positive control in the kit to enable the assay to be validated. This neutralizing antibody that we have inserted in the sVNT kit, right, is quite potent neutralizing antibodies. As of today, we haven't measured the titer for the neutralization ability, but this information will be coming very soon. So if there's an update, I'm sure that we'll be happy to share with you the information. So right now, the purpose of this positive control is really to validate your assay, so that as long as the positive control falls into a certain percentage of inhibition and OD range, then your assay can be validated. But more to come. So I'm sure the study is already being planned in our pipeline to generate the antibody neutralization titer for this positive control clone.
Okay. All right. Thank you, Dr. Lee. So the next question, customer is curious about the clinical outcomes of patients with confirmed COVID-19 infection, but with low neutralization antibodies. Are their clinical signs and symptoms more serious?
Actually, we did not include in this presentation, but we do have multiple papers coming from scientists who are using our kit, or even other papers who are using the live virus assay. So this is a great question. Do we actually believe is it people who are with asymptomatic clinical performance are having higher percentage of neutralizing antibody or lower? Actually, our hypothesis may be people who have less symptoms may have lower amount of neutralizing antibodies. However, due to the realistic and the publications, I think we have seen more than 2 of those papers. People who have less symptoms for COVID-19 actually have lower amount of neutralizing antibodies in their blood. And people who are really sick and ICU patients, they actually may have higher percentage of neutralizing antibodies. So to be, I think, connecting cPass results with the clinical performance, we are in the process of generating more data and to see what level of neutralizing antibodies may be the optimal for us to defeat against COVID-19, and we don't have that answer until today.
Okay, thank you, Dr. Lee. The next question, how do the different assays perform for patients infected with RBD mutant strains or inform on vaccines needing to protect against RBD mutant strain viruses?
Cassie, would you mind repeating this question?
Sure, no problem. How do the different assays perform for patients infected with RBD mutant strains? Or I think this is inform on vaccines needing to protect against RBD mutant strain viruses.
Damian, to your question, I don't quite get it. So you're asking different assay performance for patients who are actually infected by mutant SARS-CoV-2 strains versus the vaccine, probably some of the vaccine candidates, right? Would this be what you're asking? If that's the question, right, I believe I think there are many reported mutant strains for SARS-CoV-2, and many of them may have higher infectivity or lower infectivity due to the different mutation taking place at different locations. So I think what the cPass evaluates is not like different strands of mutations. However, we are only interested to look at individual performance of neutralizing antibody that is produced in each individual who may be able to block the RBD and ACE2 interaction. So many of the different strands, right, they may not be able to induce any of the neutralizing antibodies. And I think only when you are able to elicit a strong neutralizing antibody response to the wild type RBD domains, that can be really helping you to protect any future infection. And as you can see, majority of the vaccine candidates do adopt the wild type RBD sequences and the whole length spike proteins, etc. So I think really the purpose of vaccine is to elicit a strong immune response so that people who get vaccinated can produce a good presence of neutralization antibodies so that in any future cases, a virus that comes into circulation, when the virus have to go inside the cell, they will have to rely on the RBD and ACE2 interaction, right? So those viruses mutated, which do not already have a RBD sequences, may not be so effective because you are not able to go inside the cell and reproduce, right? So I think really the purpose, what we wanted to see for vaccine and for the general population is really to hope to see more people can come up with neutralizing antibodies so that they can protect them from future viruses that may have the potency to invade the cell. So Damia, I hope I answered your question.
Okay, thank you, Dr. Lee. I think we'll take on this last question because we are running out of time. So is the pseudovirus D wild type or G mutated type?
So the pseudovirus that we have in our product is actually a wild type RBD sequences.
Okay, thank you, Dr. Lee. So due to time constraint, we'll end the Q&A session over here. So for those questions that we didn't answer during the Q&A session just now, I'll email the answers from Dr. Lee back to you. So again, thank you, Dr. Lee, for the presentation, and also to all of you who have joined us today. I hope you find this session useful and beneficial. Yep. So thank you for joining us again, and have a nice day. Bye-bye.
Bye-bye. Thank you all.
Thanks. Bye-bye.